So I'm just going to spend time today doing a little bit more like a how-to, using the nicardipine as our example to ask questions about clinical safety and monitoring on an ongoing trial. And we'll just kind of look at some reports for safety signals. That's our primary goal here. Just as a quick reminder, this data set that we're looking at as nicardipine is a drug originally used for hypertension. It was reinvestigated back in the 80s to see if we could help ameliorate subarachnoidal hemorrhaging, so bleeding in between the brain in the skull and those tissues there. And the idea is the outcome was just kind of relieve that pressure, have an improved outcome, ultimately survival. But really, it was looking for that kind of relieved intracranial pressure. So kind of repurposing the drug, it's already been on the market. These are very sick patients, however. Once you have some arachnoidal hemorrhaging, that was your inclusion criteria, you're hospitalized, or you might have already been hospitalized. So these are a 14-day clinical trial, right? And so it's intravenous administration of the nicardipine. This is a double blind, and so the control was just placebo. And so that's where our comparator is. Here it's unblinded, so we can see all this information. It was pretty dirty. We collected things way back when from the 80s in Excel sheets and things like that, and kind of cleaned them up a little bit, made them ready, kind of this CDISC-like standard. So STTM and or ATOM are the data models that we can look at. It's highly modified. It is, again, kind of dirty. It's not perfect. That's okay. That's our intent with JMP Clinical in ongoing trial. You might have missing dates. You might have missing data. You don't have to have all those required variables or all the variables in the data model, but we just have a subset of them so you can do work. And you don't have to have all of the domains, just a subset, those that matter. So things like demographics is a minimum, and the rest is up to you. So, and you can see on our screen here what we have is we'll have adverse events, disposition, medical history. We'll have EGs, right? Labs and vital signs. We'll have some exposure information, some con meds, some study visit information. We have included the ADSL data set from ATOM, which is just more information, derived variables, along with their demographic information. And we'll merge those demographic details into kind of this metadata, superset of these things. And that's all we have. That's what we're going to look at. But it's probably important to talk about how did I register this study? So that's what we're going to do. We're going to use this nicardipine data. I've already loaded it, but I want to show you how to get started, how to get going, how to start monitoring data. So here we're in the study section here, and that's where we kind of register studies, where we kind of manipulate them. So the first thing we want, right, is to add them. So we look at a little green plus symbol. And we look for what is the name? I'm just going to type example. And I need to know where the folders of the data are, right? So we're just pointing to folders. I'm just going to use SDTM. So your biggest task will be, where is my data? Let's see if we can't hunt it down. Somewhere I have my data. Sometimes I forget where it is. And here it is. Here's our study data. Like I said, I'm just going to use this nicardipine, just the SDTM. So here it is. And as far as file types, it could be SAS7B DATs, it could be SAS transport files, or it could be JMP data sets. So that have an extension of .jmp. So you have kind of 3 file types that we can read and register and process and generate metadata about it. So we can make these reports very fast as far as responding and more appropriately arranged for you. So that's it. We're just pointing to a folder. You'll also notice that these have a file format and a file name. So we're looking for we're looking for all these data sets inside here. I'll register them, and I say okay. And pretty much that's all you need: a name and a folder location of the data sets and some basic requirements there. And as I mentioned, they have to be kind of, in this case, SDTM-like. Doesn't have to be perfect. It can be a little dirty. I don't have to follow all the controlled vocabulary that you might during submission because this is ongoing. So it's a little bit loose, but we do have some requirements, and I'll show you what happens. So I click on OK, and you can see it's going to go through, and it looks at every single one of those data sets, and it's scanning. Do I have the minimal set of data to run at least one report? that uses that kind of data. And when it's done, right, it will give you kind of a summary of what was included, what was not included. It'll give us an option to change colors and color schemes. And as Gail had mentioned, that could be an issue for some. I'm a little red-green deficient, so things get a little closer on that kind of light red and the light green scale. It's kind of hard to tell the difference between the two. So that's usually the first thing I do is I kind of switch the color schemes up a little bit. So here we go. That's kind of finalizing. It kind of gives you things in a little bit in real time. Things tend to be a little slower because we are sharing screens. Some computational resources being used just to share video and audio. Here it goes. It's finishing up. And I'm done. All right. And it'll pop up a screen here. When it lets me, I'm going to cancel out of this. I don't need to do this anymore. And this close button will be here. And you can see there it is. It's populating. Got a little screen. It's waiting for me. And there it is, all happy again. So there was the data set I added, right? Just to let you know. It also slowed down because of rerunning some things that I had in the background. We're going to look at this snapshot version. I wanted to kind of point this out. Let's make this the current study. So you can see that I have a list of studies, and then I get to choose which one I want to make the current one to evaluate. So that's what I'm going to do here. And so there we go. That's going to be the current one. And now it's it, right? As I'd mentioned, once I'm registered, you'll see up here an example. It's registered. I can go to any study, and I said, probably want to set some coloring. So there's this thing called value ordering and coloring in studies. And I can remove that lock. That's okay. So when I do this, we'll have these options, right? And like I said, for me, I'm a little red-green deficient. There is, under this color theme, something called universal. I tend to like that one a lot, especially when we get some of these light color shades. So I could use that. And then when I'm done, I can just click on apply at the bottom, or okay, it's automatically applied. And I've set that, right? So just to let people know that that option is there. So other things to let you know, sometimes I have some study preferences. And this is nice because a lot of times when we load a study, we don't want to keep resetting settings every time when I know it's very study specific. So I'm going to set those preferences here as well. And again, these could be simple things like what's my control level if I have an unblinded one, demographic subgroupings, right? I can do the analysis population. This is where the ADSL data set comes in. It tells me different patient populations. We're just going to use all subjects excluding screen failures. We could create our own age groups, things like that. So you'll see this kind of subject level information, adverse event level information. Maybe I want to do the severity stacked by severity here, or maybe by serious event or not. Let's do that. I'm going to point something out here. There is something new. The FDA has released their own medical queries. So similar to like Medro files, they're SMQs, right? Standardized Medro queries. The FDA has released their own version of them to kind of tease out those safety signals that might be lost in the preferred terms. So we can read in that Excel spreadsheet that contains that information. So this is the place to go, whether you want by default the FDA or FMQ or the standardized Medro queries, SMQ, and even versions, right? There's going to be versioning here. So this is a good place to know, like, which version am I going to set for this study, right? Because as soon as we start looking at study data, we fix ourselves on a particular Medro version or a particular FMQ version and continue that on throughout a medical query process. And so this is a way to do that, right? To fix the version. In this case, I'm leaving as newest and I only have one, right? So that gives you some ideas there. More options as you choose. And I always like to say preserve all input in case I have, some people call this SDTM plus. They add a bunch of extra columns or variables in their data sets that are not part of the SDTM data model. That's okay. I turn this on and they can be available for filtering, right? I like to use some of those additional filtering and capabilities within some of the reports. So we'll leave that. So that kind of gives you an idea. So kind of an idea of what we can do for study level preferences. And I'm done. So now that I've added a study, now that I've kind of changed some of the coloring schemes, even some of the ordering preferred the way I wanted to, I can even adjust some of the default values that I'm going to use for my reports. And I'm ready to start evaluating my data. As I'd mentioned, we're going to use a snapshot here. An important part too in evaluating our study is that sometimes we do monthly reviews. We get a new cut of the data, and we want to flag those things that are new or modified or dropped. And those could be done right here where we say add update study with the new snapshot. And so all I need to do is point to the new study data. Great. Where's the folder where the new study data exists? I click on that, and just to give you an idea, it's going to evaluate that. Here's our new snapshot, and it will do a record by record comparison to the old data that I had registered to this new data, and starts flagging, right? Flagging those rows that are new or updated or stable or unchanged. And we can take advantage of that for filtering. So really good for those month-to-month reviews if you're looking at it, or even if you're curious from a week-to-week point of view. So be aware of that one. All right. So that's what we have. That's pretty much all we do here other than delete and rename things. We do it and we're done. Well, let's go into our review section. And at this point, we can manually create a review, or we could take advantage of some of these pre-built templates. And we'll talk a little bit more about templates as well. So we could grab this medical monitoring one. That's a good place to start if you're unfamiliar with JMP Clinical. So I select it. And what will happen, it's a preset, a set of reports that we've kind of built into this template. And you can see as it's going through, it's going to go and add every single one and run them. And then it moves on to the next one and so forth, right? And so this is kind of the concept of a template. A template is a collection of reports, right? And we'll see that on the top. It will be adding a tab for every one of these things. Right now, he's building all of our patient profiles, right? That's kind of nice with this particular monitoring template. So I'm going through these 900 some subjects, right? And it's building a patient profile for every single one of these things. So it's gathering all that data, right? We can see this busy here, looking down here at the bottom, a little signal here. It's doing that. And then it's going to add each report. And you'll notice it's just not a blank. It's running it. So every time it adds a new report, it's going to automatically run it for you. And this one tends to be one of the longer ones here, right? And as I mentioned too, there's a lot going on here, so it tends to be a little bit slower than it might be for you as well. And so I'm going to pause real quick. I do see that there's some questions already. So while this is running, this is a good opportunity to answering some questions.
So one question that came up was when you're adding a study and there's some error with the data, like maybe the data is not right, is there a reporting at all or a log that tells you what the errors are?
Yeah, there is. And you'll usually get it upon the study loading. That's kind of what I was waiting for. My review kept updating as it was loading. There is a report, it's a little button, and it'll tell you what domains were included, which ones were excluded. There's even log files that are kept as well. And let me see if I can show you them. Probably worthwhile mentioning that. If we go into my documents, right, we usually see, here it is. You can see it's kind of adding things now. There's a little file under documents, a folder called JMP Clinical. And you'll see things like this default. A lot of times you'll see kind of this outputs. So sometimes things are going to be recorded there. You can see here's my nicardipine, right? Here's things that I've run on it. So here's that example of excluded variables. And that's one of their output reports. And right now it's zip code. And it's like, I don't know what that is. So I'm just, because it was empty. And so it gives you a nice little thing that I excluded this variables. So it's not an error. It's just a report. If something completely doesn't load, then it will give you an error, like it's missing something at a critical step, right? So it'll give you some similar type of report. So hopefully that'll help with this kind of processing of when you're adding a study data. And along the lines of error messages and warnings, if you happen to save a template, which again, we'll talk about this, and there's options, or there's options like set that study preferences, and it doesn't agree, the template doesn't maybe agree with what I have in the data set, it gives you a warning. It says these reports have options that cannot be satisfied. And it just says you what it's going to do. Oh, it says it can't use planned treatment, but it's going to use description of planned arm. Yep, that sounds good. I'll leave it alone. And the same thing, right? So it's all for all three of these. And I could close all of them, or I could just say, okay to close those selected reports. And I can do that. So it just tells you as that's it. So I could leave that and just say, okay. And that's basically talking to this, this demographics. We're going to go through some of these things. So the first thing I like to do in this study, again, this is a template. It's a review template. I have tabs. Each one is a report. And the first one of this one is always a good idea. A quick review of the flow of our study. So the study flow diagram or consort diagram, guys, gives you who's currently on study, right? So assessed for eligibility. I'll see that 4 were excluded, either due to some sort of screen failure or randomization failure. But then 902 were randomized. How many actually started the treatment? On some of them that didn't. And then if this is unblinded like it is here, how many went to placebo? How many went to nicardipine? And then some disposition information. How many completed? How many died? How many lost to follow? So it's a good heads up, right? As we're looking into our study and reviewing it, I always recommend that. Another one I always recommend to have is looking at the demographics as well. This is nice because now we have tabular views. And this is common, by the way, with our reports. So people not familiar with JMP Clinical, we usually have graphics and interactive graphics and interactive tables. So these are tables, listings, and figures, right? This is what we'll have them. So we have them there. I have a nice summary here by number and by percent for each of my, in this case, treatment and my unblinded. So I can see that and total. So number of males and females by sex, by race, by country, by the planned treatment, right? And so that's the key there that we're looking at. If we look in here right away, you notice that there's ability to add more. So if I had more information, or kind of like this STTM plus, which this data is kind of STTM plus, I can come back and say, all right, these are great, but I also really need to know if people had blood transfusions. I need to know if they had anticonvulsants, right? If I hold the right key down, I can select multiples. Still not holding the right key down. Come on. All right. Oh. There's a problem when you try to click without looking. All right, we'll just select them one at a time for us. We're cared about more like antihypertensive. That's the one I really care about most. But I can come back here if I get my keyboard to work. Select multiple things. And that's the idea. So if I add them, right? And we'll then throw a distribution on here. And look, right here in our tables, it automatically adds. So you can make this study specific as well. So this is a generic template. It's not study specific, but I'm starting to make it study specific. So antihypertensive, I need to know yes or no. That's good. See that balance? I see some demographic from an age point of view. And then I have my interactive components. So now I can say those that were on antihypertensives, I select them. And I get a quick review. Do I have kind of a reasonable distribution between males and females? I do see there is a bias already towards females for the study, but I just need to be aware of that. I do see that there's a little outlier immediately for this particular person, their age 108. I may want to look into their information, figure out their date of birth. It just so happens their date of birth was listed as 97, as 1997. That's not possible here. They can't be born after the study was completed, right? And ended up being this person was born in 1897, but people were using the last 2 digits for the year, right? Not surprising during that time period to save data. So one could go back and say, hey, this is valid age. Let's go correct that date of birth to be accurate. So we get like little data quality for free, but this is a real person. It's only one person that's very old. But otherwise, pretty reasonable distribution of ages. That's good. And as like I said, we can also click on our treatments as well. Nicardipine, is that kind of balanced? It seems to be pretty reasonably balanced overall. And if we looked at like demographics of age or sex, okay, that's good. And it seems reasonably balanced. So I shouldn't have too much of a bias in treatment, just knowing that there'll be more females in study than males when looking at our adverse events, keeping that in mind. The next thing I'm going to take a look at is our adverse events. Right? Let's get right to it. So here I see that a split between nicardipine and placebo. We'll see them ranked by unique subject counts. So that's what we have on our x-axis. And they're kind of ordered here based on the frequency, right, or the count of the occurrence of my particular adverse event. And these are the preferred terms. I can always go back to my data. This is kind of the default that I set for studies, but I can change that, right? I can change how it's grouped. I can say whether it's all event types or pre-treatment. I don't have to have the demographic subgrouping. I could say none. So this would behave as though it were blinded. And this would be, right, it's ordered by most commonly occurring to least commonly occurring. But if I do have things like that, why not use it? And here's that stack option. So I want to kind of maybe go to severity and intensity. Again, as I mentioned, you could set this at the study level to be more, color friendly. That was at the study preferences. You could always just come here and do a quick, ad hoc change too. And see, I'm gonna kind of keep that here. I'm gonna use my cyan for me. That stands out quite clearly to me. Other nice things that we have. This is quite a range. There's some rare events on the far right, very common things. I may want to filter this down to kind of a little bit more commonly occurring. And so we have these report filters here, right? And we can see that. And here's the overall percent occurrence. So it's set at minimum 0.1. I may want to look at 2, right? It's pretty common to see that on labels at 2% or more. So now I've narrowed it down to 2% or more that's occurring in my population. So here again, my unique subject counts. But again, if you want to show the percentage on the Y, you do that. And it kind of gives me similar information. Sometimes it's more convenient. And as I look at this, as far as those that are severe, pause my cursor, is about 9.6% severe for vasoconstriction for my nicardipine. And if I look at my placebo, I have a higher percentage of occurrence for severity. Now that should be surprising, right? Remember, this is a hypertension drug, a drug meant for hypertension. So I really should see a reduced vasoconstriction, right? I should see that. So that's good news. Drug's doing its job. Some other things as I go through, I see little blips. Here's a blip, right? And I see it's more counts overall for this one. What is this? It's phlebitis. And if I go right below, there's phlebitis down below. Oh, it's very little phlebitis. Well, it happens to be that nicardipine is known to cause, irritation of the injection or the needle site, right, where I put my intravenous infusion. So that's known. That could be an adverse event of special interest. So we know that it could occur, and it does occur. All right, not surprising. That's there. And then we can, not sure. There's some other things that it kind of scan through here. So this is one way to look for obvious signals right here. I do see a little bit right next to phlebitis. I see isothenuria. We see the system organ class here is renal and urinary disorders. And it is some issues with the kidneys. It might be more. I could just see by counts. Overall, looks like more. That's interesting. We may want to quickly drill down that. So if I come down here to my filters, I see them ordered, right? I can look for them as well. Right, here's my renal and urinary disorders. I can click on that real quick. Is there anything else? Oh, it's just the one. All right, so they're all listed here. And that looks pretty big on as far as visually. Let's look at our table view. So now I've isolated it down to my dictionary derived term. Here it is by counts and percentage, right, for mild, moderate, severe. And then we take a look at our severity and placebo. So it's definitely higher counts, right, across the board here, right? So that's pretty important. So I should probably follow up with that. Let me clear that out again. That's a pretty good signal to pay attention to. So that's good. Now there's other ways to look at this too. I like to look at this adverse event risk reporting. So this kind of I think it is a more statistical way of looking at things where we look at relative risk. So here is our preferred terms, again, on the y-axis, and they're ordered according to the relative risk or the risk difference in this case between my treatments. And we'll see that as I pause my cursor, the risk difference is 17. We'll see some confidence intervals as well. So 95% confidence intervals. So that's well above this dashed reference line. That's hypotension to be clear. I also see phlebitis and isothenuria. I actually prefer this many times over the distribution because I get a clear signal. If that's what I'm looking for, these are very clear in my perspective of looking at the difference between treatment and some reference or multiple treatments relative to our reference. So it stands out pretty clear, and even pulmonary edema. So I have some evidence here based on my forest plot, looking at risk difference, but I also can see the actual percentage of occurrence, right? And so that's important as well. Now, hypotension shouldn't be surprised, right? It's just like hypertension. I get the hypertensive drug. I should see a reduction in hypertension. So some of my treated could be more hypotensive, and that's what we see. On nicardipine, we're having more hypotensive patients. So another way to say, I can kind of know who's on the drug here, but kind of be aware of that too. So if you're not hypertensive, you're going to become hypotensive. So maybe kind of a secondary drug I need to be aware of on that. But the rest are kind of making sense. Phlebitis, isocyaninuria comes up, pulmonary edema I need to be aware of. And again, I can reduce that. If I want to look at risk difference, right, I can make it that a range. And so maybe I want to do, I'm going to say -1 to 1. Right, we're going to see that. If I did that, it's going to say, oh no, everything disappeared, but I'm going to show you a trick here. So I've done 1 to 1. And you say, wow, I filtered everything out that might be important. If you go to this red triangle, you can invert that. And that's nice because now I got an outside range, right? Or I could be more refined too, right? Maybe it's really what I want is 5. And since I've inverted it now, I can just change that. So it's a nice way to explore our values, trying to find those signals that are relative to some value or a range of values. I took advantage of the scenario to look at that. And now I've narrowed it down. So this is nice. I could see those signals that I had mentioned more clearly. And those that might be more found in placebo, right? Again, vasoconstriction, hypertension, not surprised, right? We get the sinus bradycardia, and I have a tough time pronouncing that one, so I won't. Those are on the edge of things that I may want to follow up on because they're right on the edge of what that is. What's going on here? Are they real or due to something else? Right? Now, with that said, I've said, this is great. I've narrowed it down. What if I want to narrow it down to patients? Well, it depends at this point. What kind of things do you want to look at? Do you want to quickly look at the labs because we see isocyaninuria? I may want to adjudicate that based on the laboratory data. That would be probably pretty good. I may want to look at other con meds, right, that I need to be aware of that might be causing some of things. So now it's your branch, right? Where do I want to go to this path? I think I want to go to isocyaninuria first. Let's look to see if that our labs validate what's been reported. So I'm going to go to our laboratory time trends. And here's a good way. We've just got some defaults set here. I want to change that. Let's look at some basic ones. So I'm going to remove those for a second. And it's going to rerun. As soon as I remove those, it's now going to give me a list of all labs, not just those 3. So here's all of them. So we have some of our liver functions in here, but I want to look at blood urea nitrogen. That's a pretty good indicator if there's something going on within the kidneys themselves, right? So our blood levels. And as we look at this, on average, right, and we could see our intervals here as well, on average, it's increasing over the visits. And it's kind of staying high, except for visit 12 and 13, and then jumps back up. And this is for the nicardipine. So there's some reasonable evidence based on the change, and this is not even this is change from baseline to be clear. Change from baseline is increasing over our study visits. So there's something going on, something we should pay attention to overall, right? And so we might want to filter down to just those subjects that are experiencing isocyaninuria. So that may warrant going back then to our risk report. And then we can maybe filter down. Let's filter down the rest of the reports based on that. So I'm going to go back to this, and I'm going to select all of these. And if I can do this, that's not what I want. I'm going to go back to my adverse event distribution because this is a summary view. I want a kind of a patient level summary view. So let me go back. And I'm going to go back and find that. And that's pretty easy to do. if I can scroll here slowly so I can show you those things. See my dictionary derived term right here? This is nice because I can just type in iso, right? Here's isosthenuria. If I click on that, that's another way to look for something. Here's that. And I want those that are just nicardipine only, right? I don't care what the severity is. I'm grabbing them. And what I'm going to do is I'm going to hit this little filter button, right? A little pop-up, review subject filter. I'm now going to filter all of the reports to just these patients. Now, it gives you a warning too, displaying values indicating the percent of subjects within demographic experiencing an event, right? And as soon as it's done, it's going to update with the message. There it is. Results reflective review subject filter 72, right? I have 72 subjects. So now, if I were to go back to the laboratory time trends, these are just those subjects, right? It's just those subjects that has isosthenuria, right? Again, change from baseline, it's 15%, it's kind of going up. I can go look at my con meds now. So these are the con meds that these patients are taking, right? And then with your medical knowledge, you can say, would any of these potentially cause kind of kidney function issues, right? Would that make sense, right? And I have the count here as well, how many are on there. And that's an important look at that one. I can also go back and look at now patient profiles. Let's look at all their data. So now I've added this one. I only see those subset of patients, those 72. And now I can close this window a little bit. I can add things information. I can see my adverse events over study days, right? So this is a pretty nice kind of view of things. I can say, let's go look at kind of when they start on study for this one. So that's -7, right? So I can see a little kind of pre-treatment events. And this one gets carried all the way out. So unfortunately, this subject had died at study day 91. That's outside of the actual study window, but it does kind of give you a disposition. They followed up on many of those patients as well. So I can add more information here. On the left-hand side, I'm definitely going to add things like our bun levels, right? And any other kind of things that I want to keep track of. I'll click on apply real quick. So we should see that show up. And I may actually want to, so I'm thinking about this, I may actually want to look at some of the vital signs too, right? Like blood pressure. That's a nice little indicator as well. And so now it's kind of aggregating this data. It says, oh, I didn't bring this in, so let me bring it in now. And here we go, right? So now I have my adverse event, right? Here's the isosthenuria when it began, right? It's moderate. And now here's it's isosthenuria on day 2, severe, right? And then we can see that it started and ended. You'll notice some interesting overlaps here. I think this is a data quality issue. We shouldn't have study day of moderate end at 8 when you switched it from to severe study day 2. It should have ended there, and the severe started at day 2, right? So it's always good to know that as well. Go back to the data team and say, hey, as we're monitoring this, we need to correct this. So it's not way they can have both mild, moderate, and severe all at the same time, right? So the data management team might go in and correct that. So it's a nice tool here about we're after more of a safety signal here. So that kind of aligns up with our treatment as well, right? And we'll see that it ended right there, right? So that's good. It's a good way to kind of see if anything else might be responsible. I don't think so. It doesn't look like in any of these other con meds. So I think it really is related to it. And that means I can go back to let's kind of reset all this. I can quickly go back and reset all of this. Now, I'd mentioned this. We went to the adverse events here, right? And let me reset that one as well, because this only reset my subject filter, not my term filter. Let me clear that. So we're back to the beginning. And as I'd mentioned, we have this adverse risk report. We have this distribution report. But I heard about this, this FDA, medical query. How do I add that? Because that's another way of looking for my safety signals. And you could do that. Since this is a template, that means I could add or remove. So maybe I don't want my treatment emergent. Let's talk about removing. I'm gonna remove that. Maybe I don't want that right now, but this is a good report to follow up because we have different treatment emergent adverse events segmented in different ways. I don't want my medical condition right here, like that. So let me remove that one for the time being. And I think I want some additional things I want to add. So all I have to do is look for my little bar chart here. So it'll add. We also have that same icon in the upper left. So whether you're left-oriented looking for buttons or right-oriented looking for buttons, you can add a report. It's either side. So here it is. There's the medical query risk report. That's what I want to add. So I'm going to add that. And remember, our default, we said the FDA medical queries, so the FMQs. So it's going to look for that data set, found that Excel file, and now it runs a report. Now, I also have Medra, and I'm pointing JMP Clinical to my folder of all my Medra versions. I can go back and add this again, right? I'm going to do this because I may want to compare my adverse event distribution or adverse event risk report. I want to look at the FMQ risk report, and I might want to look at that from an SMQ point of view. And I can easily switch the second one, go to the data section, and say what I want is standardized Medra queries. And I'm going to use the latest one. I have, well, not the latest one, but the latest version I have is 23.0. So that's what it's going to use. And so now I have my SMQs, right? I have my FMQs, right, listed here. And then I have my medical query, my adverse events risk report. And that's great because now I compare. Here's my preferred terms. Let's see what the FDA, their scoping of things, how they look at that. And so we have both narrow and broad terms, right? So if I just want to look at narrow real quick, I can just click on narrow in the filtering here on the left. And so I see, right, hypertension's up there, right? Heart failure, that's interesting, right? Hepatic injury, right? Hyperglycemia. I need to look at that. And if I scroll down, I'll get more details about that. What preferred terms get wrapped up into this FMQ, right? So here's hypertension. But then we see in heart failure, we have pulmonary edema. We saw that in a preferred term before, but also cardiac failure. That got rolled up into this. We see the hyperglycemia by itself, hepatic injury, right? So that I might want to look at that liver functions. I got a Hy's law. I might want to follow up with that one. Some confusional state, interesting. Would not have seen that rolled up in that way because they're very small and as far as the preferred terms. And then it's interesting. There's some bacterial infection things I may want to look at. And I go through this, right? Here's the renal and urinary infection that's separate from renal dysfunction, right? And so that gives me an idea, right? Things that might be a little different than what I expected. And so according to the FDA's medical query terms, again, from the narrow perspective, then I can switch to the broad perspective as well. And we see fall, right? Syncope and hypotension come up again from a broad point of view, even heart failure, right? Again, we'll have it in a tabular view below, right? And we'll see what preferred terms get rolled up into that kind of broad query as well. How does it go? And again, we can change this range just like it did before. I'll make this a nice range to narrow it down to those things that we think is risk difference. For any of these risk reports, by the way, if you're used to other things like log odds or logs ratios, you can change that. So the risk measurement could be odds ratio or it could be relative risk. Here we're doing risk difference by default. So that's nice. We see this. Let me close that down for a second. So we see this report. What would it look like for the SMQ, right? And so we get the same thing here. We can go for the narrow and explore. Do I get the same result? And we don't. It's a little interesting. We have cardiac failure. That's good. Hyperglycemia. So we have some nice overlap there. But tubulointestinal diseases, if I look at that one, let's see what our preferred term got rolled up. And isothenuria, right? So isothenuria got drawn out as a top one looking at SMQ, right? And all medical query risk report. So I think this is a nice way, a more holistic way, looking at multiple safety reporting, from SMQ FMQ, right? The risk report, looking at the distribution, where you really get to explore possible safety signals here. And before I leave, we saw this hepatic function and liver-related investigations. That comes up too. Well, let's take a look at our Hy's law, right? Are there any patients that might have suspected a liver injury? So looking at Hy's law here, we can see that, yes, we've got a few, right? And again, as I select these patient level points here, they're pretty severe. I could always look down at all the pairwise comparisons, and they're pretty consistent for the most part for our ALP, AST, ALT, and Billy plots, right? And then we can look at the tabular views of those as well and how I've selected them, right? And if you really wanted to, you could go back then and say, you know what, I'm going to filter these, right? Again, and then go take a look at their labs, their liver function labs, see how they've changed over time, right, for the different visits. So that's just another thing they can look at, or go look at the patient profiles as well. So now I've got, from a liver injury point of view, a suspected Hy's law. I've got my safety signals I picked up. And you know what? I've customized this report, this template. I may want to save this. And this is the last thing I want to end. We've just started the review of this data. I'm going to get some more data. I want to look at, I want to save this. I want to save the current settings as far as each one of these reports. I can go in here and click on this little floppy disk, right? Or old 3.5-inch floppy disks back in the days where we still used them. And I can save it and I can give it a name. And I'm just going to call this Nick Medical Monitoring. And I can choose where to save it. And I'll just save it where it is. And now I have it. So the next time I come in here, I'm not going to do this for time's sake. I can close this down. I have it recorded here, so I can go back, right? I can update my studies data with a new snapshot, come back to my template here and open it, and it will run everything that I have for my settings because I saved it. Since I mentioned snapshots, this usually comes up as like, how do I find those things that have changed, right? And if I'm just going to open up, I'm just going to create a new review, this little button here. Let's say a brand new template, nothing on it. Let me go to my report selector, and I'm just going to do this for adverse events. So usually this comes up in terms of adverse events. So I've got my distribution. This is all of the adverse events for my current data set. But since I did that adverse event, the comparison, I only want to show those that have changed or are new. And I could do that. If I look at the bottom, and you've seen me a little bit do this, I can go to the bottom, and there's an and button. This allows me to add an additional thing to filter by. When I scroll down a little bit again, because it went off my screen, I'm going to look for the things called review. And doing the right one? Oh, it erased it. I apologize. I was going to show it to you, and I realized I just erased that flag. It's my fault. There's something called review flag, and it would appear, and I would add it. And it would happen to be, say, like new. It would say modified and stable, and you could click on new, and this whole thing would update with those records that were new. And so, apologies, I had to refresh it this morning, and that refresh wiped out all my flags. That's my fault. I forgot to kind of re-add the snapshot to that, but that's how you would do it. You'd go add this filter here. It's called an AE review flag. When you've done your snapshot comparisons, choose it, and then you can just narrow it down to new and modified. And that's great for those monthly reviews.